So I'm going to do my best to cover units 7 through 12. Most likely, in the interest of time, if I have to cut one, the one that I will probably wind up cutting is the one on meiosis and mitosis, simply because, you know, the images for it are in your book, the pictures of the cells that you would see under the microscope are on my website, and there are countless videos on YouTube where you could essentially watch the process, see the crossover events in meiosis, see, you know, all of M phase happening in mitosis. So if we don't have the time, then I'm going to choose to skip that unit and go right into biotechnology and blood typing. So just keep that in mind. If that unit's missing, that's the reason why. There's just enough resources, and I don't want to waste time that could be spent on some of these other techniques. But of course, we don't have to worry about that until we get closer to the end of this lecture. The first unit I'd like to cover today is going to be the one that you did immediately after the break, which was the one on diffusion. Okay, so when talking about diffusion, remember what we're really talking about is we're talking about the movement of molecules or ions from an area of high concentration to an area of low concentration. And we call this difference in concentrations a concentration gradient. So we always see ions or molecules diffusing down their concentration gradient. The only time we ever see them going up it, which we didn't cover in lab, was during the instance of active transport. Okay? Now, there's one other type of diffusion that you also learned about during this lab, and that particular type of diffusion was called osmosis. Remember that osmosis is simply the diffusion of water across a selectively permeable membrane. Right? So, any general questions right now regarding diffusion or osmosis? The discrepancy between the two? Pretty basic stuff. Okay, so before we get into the thick of all of the experiments that were performed in this particular lab, let's start with the very first experiment, which was simply devised to demonstrate factors that can ultimately affect the rate of diffusion. So you may recall that in this experiment you had two plates. And you took these two plates and you created these circular cavities in them. And you added two different substances to these plates. In one of these cavities, you placed a substance called Janus green, which ironically enough was not actually green. Okay, but for all intents and purposes, we'll say that green here equals Janus green. Okay. And then in the other one, you put a substance known as potassium permanganate. Now, does anybody remember what the difference was in mass or molecular weight of these two dyes? Does anybody, I guess, remember which one was the heavier one? So I believe it should have actually been Janus green, right? So Janus green was the heavier one from a molecular weight standpoint. So there were two things we were going to show. Okay? We were going to show that molecular weight and or the size of a molecule has an effect on the rate of diffusion. And then you may also remember we put these two plates at different temperatures. One of these plates was simply left out on the bench top at room temperature, which is approximately 25 to 27 degrees Celsius, depending upon what the situation was in your lab. The other was placed in an incubator at 37 degrees Celsius. So, the two things we're testing for here are which of these two dyes diffuses faster, and then when we place the plates at different temperatures, will we see a difference in the rate of diffusion of the same dye? Questions, concerns so far? Everybody good? Awesome. So, which do you think is going to have an easier time diffusing? A heavier molecule or a lighter molecule? A lighter molecule. Now, the way that you observed this was in the halo that resulted around the cavity. The lighter of the two molecules wound up making a larger halo around its respective cavity. So Janus green, if I'm not mistaken, only had this tiny, tiny halo of dye around its cavity, while potassium permanganate had this huge ring of dye around its cavity, signifying that the lighter of these two molecules is going to diffuse faster through that semi-permeable agar that made up that plate. Any questions so far? Okay. Now again, we expect to see the same result on this plate, but there's one other piece of information we're going to get from this plate here. And the question we're really asking between these two plates, so on each individual plate we're asking, does Janus green diffuse faster or does potassium permanganate? But when we look at these two plates, there's a question we're asking when we're comparing them. And that is, when do we expect to see potassium permanganate diffuse fastest? At room temperature or at 37 degrees Celsius? So if room temperature is about 25 to 27 degrees Celsius, and this is 37 degrees Celsius, where do I expect to see the fastest rate of diffusion for potassium permanganate? 37. Somebody justify why. What does an increase in temperature typically mean for kinetic energy, the energy of motion? It means an increase in kinetic energy. Faster moving molecules are going to diffuse faster. So what result do I expect to see in terms of the halo here compared to the halo here? Bigger. I expect to see a larger halo here around this potassium permanganate than I did over here, signifying that the rate of diffusion on this plate happened faster as a result of the increase in temperature. And I might also see the same thing happen for Janus green. So a little bit larger, not too much larger, but large enough. And again, notice that I still have the consistent results I saw on this plate. That is, potassium permanganate still diffuses faster than Janus green. Questions about the plate exercise. So this demonstrated two factors that have an effect on the relative rate of diffusion: temperature and molecular weight. Okay? All right. So now let's go ahead and take it a step further. I want to talk to you about the remaining experiments that you performed in this lab. But to do that, we have to establish one other thing. So we were looking throughout the rest of the lab at osmosis. And in one instance, we did look at selective permeability. Okay, and we'll talk about that here in a moment. But for the most part, in 3 of the experiments in this lab, we were looking at osmosis. And you may recall from the textbook that the textbook stated that in the case of osmosis, you will always see water diffuse from the area of the lowest solute concentration to the area of the highest solute concentration. And so what I'd like to talk to you about now is I'd like to talk to you about terms that we use to differentiate solute concentrations between two solutions. Now my lecture students, you've already heard this joke, so you don't have to laugh to placate me. But we'll see if the rest of you choose to. All right? So, to determine or discuss the difference in solute concentrations between two solutions, we have to expand our vocabulary. So let's go ahead and expand that vocabulary now. We're going to do so by first implementing a root word, tonicity, or it sometimes also is just written simply as tonic. Now, tonicity is solute concentration. It's got 3 prefixes that have the capacity to be in front of it. One of these prefixes implies that the solute concentration is higher in one solution compared to another. One means that it's the same, between one solution and another, and one means that the solute concentration is lower in one solution compared to another. So let's go ahead, my students especially, let's start with the most obvious. What prefix have we seen again and again this semester that always means equal or the same? Iso. So anytime you see iso, that means the same or similar. So an isotonic solution is a solution that has the same solute concentration as another solution. Okay. All right. Now, you go to a daycare center, you see the one kid who's got way more energy than anybody else swinging from the rafters while the other kids are at nap time. What is a term you typically use to describe a child who has a higher level of energy than any other child? Hyper. Hyper. So, hyper means higher. Okay? So, a hypertonic solution has a higher solute concentration than another solution. And finally, let's say instead you have a lower solute concentration. Now, those of you who have worked in the healthcare field have probably experienced this. If you've interacted with someone who has diabetes, if they get to a point when they have low blood sugar, what is a term that defines that? Hypoglycemic. Hypo means low or lower. So a hypotonic solution is one where we have a lower solute concentration than another solution. Now here's a very important point that I want to bring to your attention, because a lot of students don't use this term correctly. They'll just be given an arbitrary solute, or I'm sorry, solution, and they'll say, this solution is hypertonic. You can't use it that way, okay? That's like me coming in here and saying, I'm taller. It doesn't make sense. I'm taller than what? Okay, the answer, of course, nothing, but that's okay. Instead, I would have to say, I am taller than nothing, or I am shorter than everyone. Okay, so these are comparative terms. You have to have two solutions if you're going to use the terms iso, hyper, or hypotonic. You're comparing that solution to another solution. If you don't have two solutions, you can't use these terms. Okay? Any questions or things I can clarify right off the bat in regards to tonicity? Okay, so let's start talking about what happens with water whenever we are using solutions of varying tonicity. Now there's a very simple thing that you can always kind of keep at the forefront of your mind about water, and that is that water always moves towards a hypertonic solution. Okay? So water always moves towards a hypertonic solution. So if you have a certain arrow showing you water moving in one direction, what you can pretty much know is that water will always be moving away from the hypotonic solution and towards the hypertonic solution. And it doesn't matter if the arrow is going one way or another. If water is moving in a single direction, that is, osmosis is only occurring in one direction, that water is always moving from the hypotonic solution towards the hypertonic. And I'll justify to you why this is when we get into one of the later experiments. If instead you have an instance where water is moving in both directions at an equal rate, then both of your solutions should be isotonic to one another. Okay? So this is just the general parameter of what you can expect for your solutions if water's moving in one direction or another. We'll talk about the effect that this has on cells soon enough. Okay? All right, so let's go ahead and jump into experiment 8.2. And you'll recall with 8.2, what you were ultimately doing was you were setting up these beaker and bag combinations, where the beaker or bag could contain either a sucrose solution or it could contain water. So, let's go ahead and draw out our three beakers. All right, so we've got our 3 beakers. I'm going to place bags into each of these beakers. Okay, and in the first, I believe it was water in the bag. In the second, I believe it was 10% sucrose in the bag. And in the third example, I believe it was also 10% sucrose that was in the bag. And then, of course, the solution that we had outside of that differed as well depending upon which one we were looking at. So in this first example, we had 10% sucrose solution in the beaker. In the second one, we had water in the beaker. And then in the third one, we had 10% sucrose in the beaker. Now, what I always like to do personally when I encounter a problem like this is rather than trying to look at the diagram, I always like to draw this out for myself. Because it makes it a lot easier if I can draw and I can kind of follow where I think the water is going. And right now, they're not telling you where the water is going. And if they give you a problem like this, they're probably going to ask you, where is the water moving? Okay, now, a word to the wise, please don't write a one-word answer. Okay? On past practicals, there have occasionally been questions like this, where it asks, where will the water move? And some students write the word up. Okay? Up doesn't mean anything. You have to say, is it going out of the bag, or is it going into the bag? Or is it going out of the bag and into the beaker, or out of the beaker and into the bag? Don't just write out or in either. Because I can say that water is going into the bag and out of the beaker, or I can also say that water is going out of the bag and into the beaker. So you can't just write the word in either. You have to specifically say, water is moving from the beaker to the bag, or water is moving from the bag to the beaker. Okay? So if they ask you a question like that, please make sure that you're as specific as possible. Again, you're not writing a great American novel. You should be able to answer all these questions in 5 words or less in most instances. But you want to be specific enough so that your instructor knows what you mean. All right? So, what I'm going to do, I'm going to show you how I would approach this, and then if you want to approach it the same way on the practical, feel free to. If not, then hey, have your own way, that's fine. What I like to do is I always like to break down bag versus beaker, because it gives me a clearer idea of what's actually happening. So I always write out bag, beaker, and I draw my dotted line representing the membrane between the two. Now, these membranes, remember, are selectively permeable. Water can move through them, but sucrose cannot. Okay? So, in the bag, let's say, well, let's not say, we have it. We have water, and then in theory, we may or may not have sucrose. We'll talk about that here in a second. And then here in the beaker, we have 10% sucrose. Okay? Now, this is 10% sucrose solution. So I have a very fundamental question for you. If 10% of the sucrose is, or I'm sorry, if 10% of the solution is sucrose, what is the rest of it? Water. So what percentage of this solution is water? 90%. All right, over here, I have water, pure water. So in theory, what percentage of my solution here is water? 100%. So does that leave any room for any sucrose? No, it is 0%. All right, now we've already established that sucrose can't diffuse anywhere because this membrane is selectively permeable. So what is the only thing that is going to diffuse in this particular problem? Water. And where is it going to go? From the bag to the beaker. Remember, water is like any other molecule. It goes from the highest water concentration to the lowest water concentration. The highest water concentration, 100%, is in the bag. So water is going to leave the bag and go into the beaker. And so that would look like this. Okay. We'll come back to that in just a second. We'll define our hyper and hypotonic solutions. But I'd like to move on to the next example. So, if we were to do the exact same thing here, we draw it all out, we would just find that we reverse these two things. So now, my 100% water is going to be in the beaker, My 90% water, 10% sucrose, those are going to be in the bag. So where is water going to go in this second example? It's going to go from the beaker to the bag. Excellent. Okay? And then finally, here, in this third example, I find that I have 10% sucrose and 10% sucrose. So that means both solutions are going to have 90% water, which means is water going to go into the bag, out of the bag, or in and out at the same rate? It's going to go in and out of the bag at the same rate. Excellent. Okay, so we've established now where our water is going. Now what I'd like to do is I'd like to ask a few follow-up questions. Now the main thing we were trying to determine in this first exercise was, is the bag gaining mass, losing mass, or staying the same mass? So in this first example, the bag is losing water. What effect is that going to have on the mass? It's going to decrease. So in this first example, mass decreases. Okay? In the second example, water is moving into the bag. What effect is that going to have on the bag's mass? It's going to increase. Excellent. Mass is going to increase. And finally, in this third example, water is moving in and out at equal rates, so what's going to happen to the mass? So mass is going to be unchanged. It stays the same. Good. All right. Excellent. So now, let's look at each of these solutions surrounding the bag, and let's determine if those solutions are hypertonic, hypotonic, or isotonic. Okay. So, this first solution here in the beaker, okay, hypertonic, hypotonic, or isotonic? Hypertonic. Two ways you can tell this. First and foremost, hypertonic means higher level of solute. Solute is your sugar. We do have a higher level of solute here in the beaker, meaning that that is hypertonic. But the other way you can tell it is based off of what I drew for you here. Water always moves towards the hypertonic solution. The arrowhead always points at the hypertonic solution, and that's exactly what this water's arrowhead is doing. So this is going to be a hypertonic solution compared to the solution in the bag. What can you tell me about the tonicity of the bag solution compared to the beakers? It's hypotonic. Whenever you have a hypertonic solution, the other solution is always going to be hypotonic, and vice versa. Okay? All right, so let's move on to the second example. Describe this solution for me in the beaker. Hypotonic. Excellent. And what does that mean the tonicity is going to be of the solution in the bag? Hypertonic compared to that of the beaker. Excellent. And finally, here in this final beaker, what do we have? We have an isotonic solution. Excellent. All right. Any questions about experiment? I guess it was 7.2? Okay. So experiment 7.2, no questions? All right. So let's apply these fundamental characteristics to plant and animal cells. Okay? Because we didn't just do this with bags, you actually placed animal and plant cells in these different types of solutions. So let's go ahead, we're going to draw out our test tubes for animal and plant cells. We'll have all of it up here together so we can kind of compare and see if anything unexpected happens. All right, so these are going to be my animal cells, and these are going to be my plant cells. Now, you placed your animal cells into solutions that were isotonic to the cell, hypertonic to the cell, and hypotonic to the cell. So, first one, I placed a red blood cell into an isotonic solution. First question, what happens with water? Moving into the cell, out of the cell, or in and out at the same rates? Same rates, in and out. Excellent. The water goes in and out. Follow-up question, is that going to have any effect on the cell's morphology, the cell's shape? No, they're going to be these nice round erythrocytes, just as I have drawn there. Excellent. Okay? Now, I place a red blood cell into a hypertonic solution. Now, what effect did we see happen with the bag when we placed it into a hypertonic solution? It lost mass. Water left that bag. Well, water is going to leave the cell as well. Now, with red blood cells, what is the outermost structure on an animal cell? The cell or plasma membrane. So that water leaving represents a decrease in water pressure, and what that does to the cell is it causes the cell to collapse upon itself. So it takes on this shriveled kind of state because water is leaving the cell. Okay. Excellent. And finally, I place a red blood cell into a hypotonic solution. Now, what did we see happen with the bag when we added it to a hypotonic solution? It increased in mass. Now, that represents water going into the bag, or in this case, into the cell, causing the cell to swell. Now, that plasma membrane, is it a rigid or a flexible structure? Flexible. Can it handle a whole lot of water pressure? No. So, this cell literally bursts. So water moves into the cell, and that causes the cell to burst. And we call or refer to this state or this process as lysis. The cell itself is lysed. It has effectively burst. Good. Questions, concerns? Pictures of those on the website, so definitely look at them if you're having trouble remembering what each of these states look like. By the way, you're not going to see anything here because all your cells have exploded. So, if you're wondering why I don't have a hypotonic slide, that's why. There's nothing to see. All right, now let's talk plants. Now in plants, we added them to either a hypotonic solution or a hypertonic solution. So we'll start with hypo. So I added my plant cell to a hypotonic solution. And what I probably saw here in my plant cell was I saw all of the chloroplasts evenly dispersed through that plant cell. Now, if I place that plant cell into a hypotonic solution, water is going to do the exact same thing that it did in my bag and in my red blood cell. So where's water going to move? Is it going to go into the cell, out of the cell, or in and out at the same rate? It's going to go into the cell. Now, a quick question. Why do plant cells not burst then? The cell wall. Does anybody remember what we call this state when a plant cell is really, really swollen with a lot? Yep, turgid, exactly. A lot of water pressure, that plant cell exists in what we call a turgid state. That happens when you place a plant cell into a hypotonic solution. All right, so now what I'm going to do is I'm going to place my plant cell into a hypertonic solution. Now, does the cell wall collapse when that happens? No, but what does collapse? The plasma membrane collapses. And when the plasma membrane collapses, it brings all of the organelles to the center of the cell. You probably saw this, a bunch of the chloroplasts kind of all jam-packed towards the center of the cell. We have a word to describe this, and that is plasmolyzed. And this also means, if I'm placing it in a hypotonic solution, that water is moving where? Out of the cell. Water is leaving the cell, and that change in water pressure is what causes that plasma membrane to collapse. Okay. Questions, concerns on 7.2 or 7.4 and 5? All right, let's talk about the very last of the experiments that you performed in the diffusion lab. This one was intended to test selective permeability. So, in this, what you did was you had a mixture. And in this mixture, you had several different things. mixture you had sodium chloride, sodium sulfate, glucose, starch, and protein. So this mixture was in a bag, and you had a selectively permeable membrane that constituted the outside of that bag. And the question you're really asking in this experiment is, which of these molecules are small enough to diffuse across that bag? And so over here we have the beaker, and this is where you drew up your solution as a means of testing for each of these. So, truth be told, what you were really testing for here were chloride ions, sulfate ions, glucose, starch, and protein. Now, if only small molecules are going to be able to permeate this bag, and we've already established in the previous experiment that sucrose, which is a disaccharide, cannot permeate the bag, there are actually two molecules right off the bat I know should not diffuse across this membrane. What are they? Starch and protein. Glucose is only a monosaccharide, not a disaccharide. That's why I can't immediately say that it should not be able to make it across. Starch, however, is a polysaccharide, and most proteins are larger than disaccharides. So I don't expect these two to be able to diffuse across the membrane, but just to be sure, we still tested for them. Now, does anybody remember way back eons ago, like 3 months, what we used to test for starch and proteins? Iodine for starch. And then what for proteins? Biotin. Excellent. And of course, when we tested for both of those, hopefully they came up negative, because those are too large to diffuse across that bag's membrane. Good. All right, so let's talk about the remaining ones. Now, I think personally, based off of the idea that sucrose can't diffuse across the membrane, the one that's the most questionable here was glucose. Okay? But these two, as you can probably see, this one's only a single atom or ion, chloride, and this one's only composed of 5 atoms. Sulfur, oxygen, well, technically 4 oxygens. If you're wondering why I'm saying this, it's because when we place both of these solutions in water, the sodium has the tendency to dissociate from the chlorine or the sulfur and oxygens. Okay? So, these two were small enough to where they did diffuse across the membrane, but we still have to test to ensure that they're there. So does anybody remember what the detection agent was that we used to test for chloride ions? Silver nitrate. If you get confused and you say barium chloride, think about it. Would it make sense to test for chloride ions with something that has chloride already in it? No. So we're going to use silver nitrate. Now, quick question, anybody remember what the positive coloration of this was? It was like a white or gray color, right? It was white and cloudy. So that's the positive. Anybody remember what the negative was? The negative would be clear. All right. With, not with silver nitrate, but with sulfate, we also needed to test for the presence of this, so we used the solution called barium chloride. Does anybody remember what the positive color here was for that? It was also a cloudy white. Anybody want to take a stab at what they think the negative is going to be? Clear. Good. Awesome. And then that just leaves glucose. Now, depending upon the bag that you got, I believe the bags that are here are supposed to be large enough to allow monosaccharides through. Gesundheit, okay? But just barely. So you probably got a slight positive for glucose. Now somebody remind me, what solution do you use to test for the presence of reducing sugars like glucose? Benedict's. And what is always the key step that you have to do if you want to get any kind of result? You have to boil that. Good. So you probably got a slight positive. It was kind of a greenish brown that lets you know that there was glucose there. Questions, concerns about the selective permeability experiment? If you didn't see glucose, that's okay. Glucose is, again, much larger than these other two. And again, if at your particular campus they got a different kind of bag, glucose might have been too big to permeate it. So, any other questions, concerns? All right, so that brings us to the end of diffusion.